G'day guys. This is the long-awaited sort of video that I wanted to do on the gut microbiome, and sort of will sort of elucidate or explain the importance of an ancestral diet in restoring proper gut function, and why an ancestral diet is the only way of actually achieving this, and that other approaches do not work, simply do not work, because they don't deal with the prime regulator that's involved in this, and they ignore the important peptides that are in basically animal products that have this effect. Let's get started. I'll just share my screen. This is a study that was done a while back. It was actually published on 2015 of December in Cell. Cell is a quite a prestigious magazine in terms of doing hardcore. It primarily looks at hardcore science, things like cell metabolism, immunology. It's more, they are more focused on looking at mechanisms and stuff like that. It's the quality that you'll find in Cell is much higher than many other publications out there. You don't see a lot of epidemiological nonsense in Cell. It's a much more hardcore terms of biology, physiology, biochemistry, those sort of things. So we won't go into the full, we won't look at everything in this. It's more to go through the abstract, cover a few points. And if somebody actually is interested in actually going right through it and actually looking at more detail, go ahead and enjoy yourself. In that regard, I say most people just want to get the main facts, without going too much heavily into the biochemistry. But sometimes we do have to cover these sort of studies, because it's the only way we can really understand what's actually happening inside us or in organisms. Microbiota, modulated metabolites. That's what we're going to be talking about, metabolites that are involved in basically the shape of the intestinal microenvironment by regulating NLRP6 inflammasome signaling. So this is key inflammasome that is involved in that. There are other ones. There's also the NLRP6/3 inflammasome, and I will cover that shortly. As you can see it right up there, and we'll get into it. So this study was done with a number of different organizations, primarily in Israel. There was some participation from Bonn University in Germany, and from also the Yale University School of Medicine in the US. Now let's get into it. Host microbiome co-evolution drives homeostasis and disease susceptibility. So when we're talking about here is between the host and the little buggers that are inside, there is a co-evolution, an interaction, and we sort of cohabitate that in that space, our gut space anyway. And we're looking at sort of things that control homeostasis and things that derange things, and disease susceptibility. Yet, regulatory principles governing the integrated intestinal host commensal microenvironment remains obscure. While inflammasome signaling participates in these interactions, its activators and microbiome modulating mechanisms are unknown. So, and that's been this, a lot of researchers just looked at the associational relationships to dysbiosis, irritable bowel syndrome, and all the other sort of stuff. And it's really been very poor in sort of identifying the real mechanisms behind this. Here we demonstrate that the microbiota associated metabolites, taurine, histamine, and spermine, shape the host microbiome interface by co-modulating NLRP6 inflammasome signaling, epithelial interleukin-18 secretion, and downstream anti-microbial peptide profiles. So when we talk about peptide, we're talking about small, 5 to 20 chain long amino acid, little protein systems that do interact in a certain way within our system. We're full of micro machines, remember. That's just another one of them. Distortion of this balance, AMP landscape by inflammasome deficiency. So when you get this balance of the AMP landscape by inflammasome deficiency, so when we don't have enough, drives dysbiosis development. I wonder what is required to actually have good functioning NLRP6 inflammasome. Distortion, upon fecal transfer, colitis-induced microbiota hijacks this microenvironment, orchestrating machinery through metabolite-mediated inflammasome suppression. So some of these nasties basically hijack the system and suppress NLRP6 inflammasome signaling, leading to distorted AMP balance. These are basically AMP, as I said earlier, these are also affecting the innate immune system, which is very important to understand. Favoring its preferential colonization. So when you suppress this, you get some nasties that can actually colonize and derange things and allow other things to increase, like histamine. Exactly. Restoration of the metabolite inflammasome AMP axis reinstates a normal microbiota and ameliorates, resolves colitis. So as you can see, if you can actually restore the proper functioning of inflammasome NLRP6 AMP axis and balancing AMP, then your microbiota actually starts restoring back to normal, rebalances itself. So do I need to take probiotics? Now, I do occasionally recommend probiotics for certain things to basically, as interventions, early interventions, to try and actually suppress certain pathogenics so we can get things reset, and reset. And some of these actually also play an important role, if you want to basically be able to, they have an important role in sort of keeping under control those pathogenic bacteria. But the thing that actually makes sure that these things start happening and the right bacteria are supported, and the ones that are not so good, are sort of down-regulated but basically kept under control, is this basically key inflammasome AMP axis. Really, really important that we sort this out and we restore normal microbiota functioning. This is what they're seeing with this research. You can actually cause and effect mechanism. We can put a metabolite in, it can correct this axis, and things go back to normal. That is cause and effect, and you can do it over and over again. That is mechanistic science, which shows a causal relationship between certain metabolites. We'll go into it shortly, that it basically sort this out. Therefore, targeted postbiotic metabolomic interventions may restore a normal microenvironment as a treatment or prevention of dysbiosis-driven disease. Now, obviously, the best results would be to also try and actually restore some of the beneficials in there to improve the general population. While this metabolite, this chematologist we're going to be talking, does have that role, but as well to give you a more, you could say, well, it may resolve these issues. At the same time, we still want to have good digestion to be able to deal with certain types of foods that we have lost the capacity to digest, like especially the loss of a lot of lactobacillus and stuff like that. So there is still an important thing, but when you actually modulate that, it will preferentially support these beneficial bacteria like lactobacillus and bifido-infantis and all that. They will increase. They will be preferentially looked after when you've brought the gut microbiome with these metabolites we're going to talk about back into homeostasis. Got it? Okay, now let's look at the graphical abstraction. So rather than going through a whole lot of biochemistry, which would bore the living daylights out of everyone, we'll just look at the graphical representation to see what is happening, which makes it easier. As you can see, there is a lot of pages in this, so it would take quite a long time. Inflammasome, AMP, microbiota, and metabolites. I wonder what these are. So we're talking about taurine, histamine, spermine, indigenous microbiota, signal, LPS, and second signal metabolites, which are these. And there is AMPs. So you do want a bit more AMPs because they are actually going to be playing a role here. Now, as you can see, the more taurine you've got, NLRP6 inflammasome actually works, which means that you get more interleukins, which are part of the innate immune system. And they can actually, with AMP, actually work to make sure that we've got a good microbiome. When you shut off this completely due to a lack of taurine, or when we say taurine, we mean animal nutrition, period. What you do is you shut this down, and basically you actually, interleukin 18 declines, less support for AMPs, you get a derangement, a dysbiotic environment. Host genetic deficiency leads to, so if you genetically, deliberately turn off, they have basically what they call in this, we're looking at knocking out of these ones that are deficient. We see this, and also we see, mice deficient in NLRP6, and we look at dysbiosis that drives potential intestinal autoinflammation, inflammation-induced colorectal cancer, and features of the metabolic syndrome. So we know what basically it is postulated the factors secreted by WT microbiota activate, modulate this, but no such factor has been discovered to date Moreover, the mechanism by which inflammasome deficiency leads to dysbiosis and how this disease assumes dominant upon transfer host remains elusive. In this study, we highlighted principles by which the host and its microbiota cooperatively utilize inflammasome signaling in the formation and stabilization of host microbiota interface. We identified, so that's the general stuff that what other studies have been talking about. And then we're talking about down here what this study, we had identified microbial metabolites that signal to NLRP6 inflammasome, thereby activating host immune circuits to orchestrate an anti-microbial program, which optimizes commensal colonization and persistence. So you actually get these systems. This is where the immune system is actually modulated. This is where taurine is modulating the immune system in the gut to basically, through NLRP6 inflammasome, and you're actually getting, a reorganization, rebalancing of the commensal bacteria in the ones that in the right sort of way. We demonstrate that alterations of this crosstalk is sufficient to induce a state of dysbiosis. We demonstrate that the dysbiosis and that altered microbiota is able to exploit these pathways in conferring a preferential microenvironment for its colonization in an invaded host. So basically, they've basically used these sort of they tend to use in the absence of germ-free mice results in, without going into two. Let me just find the other part. And basically to determine whether the reduction of intestinal interleukin-18 and AMP levels is responsible for the inability of inflammasome deficient mice to develop a normal microbial community structure. We demonstrated this blah, blah, blah. Mice that determined the change in bacterial composition. So they noticed that when that wasn't there, they noticed that was actually happening. So, the reductions in inflammasome signaling and interleukin-18 production. Mice was accompanied by alterations in the global antimicrobial transcription program of the colonic mucosa, inducing a reduction. These continued interleukin-18 replenishment of recipient mice throughout the co-host period, partially prevented dominant transmission of dysbiotic microbiota configuration from inflammasome deficient mice. So they basically put in interleukin-18, so artificially, and that sort of prevented this dominant change into pathogenic sort of deranged microbiome. Together, these findings demonstrate that the dysbiotic microbiome stemming from inflammasome deficiency. Mice modulates inflammasome signaling upon transfer of new W host, the consequence of anti-microbial melee. The new colonization microenvironment changes and resembles a dysbiotic niche of origin, allows for community persistence of invading microbiome. So things that shouldn't be there invading. Now, let me just go. to this study. Okay, so therefore, a surprising finding was associated with Crohn's disease susceptibility with NLRP3, which is the other signaling, or interleukin-18 gene polymorphism. This has to do with the genes, known to decrease. So if you decrease this in the inflammasome or the interleukin-18 expression, subsequently several groups reported that these sort of deficient mice develop more severe sort of colitis, dysbiosis and stuff like that due to the rupture of the intestinal barrier. So we're talking about damage to the intestinal barrier. Now we've looked at the other previous study that I did. I'll put it up here, that we looked at with which was the taurine one that showed, you know, the tight junctions, you know. So taurine's playing a lot of roles in everywhere along through here, but when there's not enough and inflammasome is low, you're going to get these ruptures in the gut barrier, you know, which ain't good, associated with proliferation of commensal bacteria, increased chemokine production and leukocyte infiltration in the colon, yes, and damaging that colon. These phenomena are due to decreased interleukin-18 more than interleukin-1b production in intestinal epithelial cells, and can be reversed by injecting what we saw in the previous study, injecting you know, the stuff inside can be reversed of the injection. Yep. Interleukin-18. And then these are just the studies, 79, 80, you know, down there, links. So they should have put a space there because it looks like it's connected with this interleukin-18 injection. Another, which we saw in the previous one, another inflammasome, NLRP6, which we looked at before in the other study, is highly expressed in the large and small intestine, especially in the intestinal epithelials, and appears to be involved in interleukin-18. Both these are. Here's LFRP3 inflammasome is a critical component of the innate immune system that mediates capsicum 1 activation in the secretion of pro-inflammatory cytokine interleukin-1b / interleukin-18 in response to microbial infection and cellular damage. So when there's cellular damage caused by these nasties, or these nasties are invading an infection of some sort, interleukin-18 gets out there, sorts it out, clears it out, okay? Basically, your innate immunity, just for those people that don't know, is your first line of defense against germs entering the body. Got it? You need your first line. I mean, I've talked about T cells and all that for viruses and all that. But you need your first line of defense. These are different components of your first line of defense of your innate immunity. And if you don't have that, you know, supercharged with vitamin D and taurine, you're going to be in trouble. Simple as that. Meat and sun, basically, or have a steak out in the sun for good gut health, like our ancestors did. So just for people to know what interleukin-18 is, interleukin-18 can modulate the innate and adaptive immune system, and its dysregulation can cause autoimmune or inflammatory diseases. So you don't want this to be too out of control. That's where vitamin D is modulating these things. That's why it's important to have vitamin D in the picture as well, because it has a modulating effect. Got it? So we need that sort of stuff. Plus also taurine has a modulating effect as well. So getting back to here. To the graphical part. As you can see, when we've got too much histamine and spermine. Now, spermine is fine to be in your reproductive system, but we don't want too much in the gut, okay? And we don't want too much histamine in the gut. And that usually happens when you don't have enough taurine. So all these blood, and that's when this happens, this microbiota transfer, which actually increases histamine, increases spermine in the actual, because spermine actually acts as, just to basically cover this, spermine is I won't go into it, polyamine involved in cellular metabolism and can be found in eukaryotic cells. That's fine. And basically in your reproductive cells, not a problem. The precursor for synthesis of spermine is an amino acid ornithine. It's an essential growth factor in some bacteria, got it, as well. So in the gut, it's actually acting as a growth factor for those bacteria. That we do not want. We want it properly regulated, not things deranged, out of control. And that's how we're also getting SIBO. Like, due to spermine, we're actually getting too much growth, and we're ending up with SIBO, with small intestinal bacterial overgrowth. Got it? So that's the derangements that are happening when you're not on a species-appropriate diet. So inflammasome is being blocked, and the amount of AMPs is very small, and the amount of interleukin response is very small. So basically, you're in a mess. You're in dysbiosis. You put taurine back in, it suppresses these other players and basically supports, see taurine blue, taurine supports inflammasome, gets it going, gets it all working, pushes up with inflammasome. You can actually increase interleukin 18, AMPs. It eliminates the problem and restores normal gut microbiome status back to, here, if you continue in such a poor diet, by completely blocking this, you basically end up with a hijacked system in a mess. That's what you end up. You throw in a big juicy steak. So when you hear people in the low carb community say, oh, don't eat too much meat. And then they wonder why a lot of them who are basically starting to create a more plant-based orientated, low carb ketogenic diet, why they still have a lot of gut issues. Well, it's simple. They're not getting enough taurine to fix it. That's why, that is part of the problem. Now, also, let's continue with this. So NLRP is involved in this. So we talked about another inflammasome, NLRP6, which we've covered, is highly expressed in epithelials, and involved in interleukin 18 production in the digestive tract. I lost my spot. NLRP6 is involved in the response to viral pathogens and bacterial components of the commensal microbiome. Microbiome-derived modulator of NLRP6 activity has been identified. Now we're finding out what the identified thing, such as the bile acid derivative taurine. Got it? See, taurine actually makes bile salts that stay in the intestine for much longer. They have a role there to play in controlling that microbiome. Got it? As a positive regulator, got it? Positive regulator. It's not, maybe not. No, you put it in, you get a positive response. You take it out, you get a negative response. That's basically fulfills mechanism in terms of inference of causality. A regulator of interleukin 18 production, or spermine and histamine as a negative regulators. So spermine and histamine are negative regulators. So when there's too much of them, you need some histamine and you need some spermine in there, but only some. When there's too much, that's when you're in trouble, you know? So, because you still got turnover of the gut microbiome, your stuff like that, and you still need a bit of support there to support, and you need a bit of history because it actually does play an important signaling role as well. And actually, you know, in a smaller amount, it has certain benefits, which I'll go into another video. I'm not going to cover it in this video, but when it's too much, that's when it's in a deranged state, that's when it's a problem. NLRP6 activation induces protective mucose production by goblet cells, as well as interleukin-18 dependent antimicrobial peptides, which is the AMPs. Got it? Increases AMPs, which basically keep down the nasties, which we saw in the graphical representation by the epithelial cells, and play a role in the maintenance of normal microbiota in the lumen. Indeed, NLRP6 knockout mice develop, so when you knock out inflammasome, which, remember here, that's like knocking it out, that's completely closing it down. When you knock out, developed a dysbiotic microbiota. Surprise, surprise. Notably increased of prevotellins. Our dear, you know what, Prevotella that all the vegans talk about. It's one of the actual part of the family, the genus Prevotella. Now, Prevotella has its role in the gut, but excessive amount, and I've done another video on that. I'll place it somewhere down there in that regard, but it's part of my myths and falsehoods of veganism, which shows a sort of inflammation that actually can induce when it's in very high amounts. Basically, it goes up quite a bit. Surprise, surprise. On a veganerized diet, it goes very high, which is not a good thing, and suppresses some of the other important, you know, it changes from the bifidobacteria type to more Prevotella, which isn't a good thing. Anyway, so by fixing inflammasome, we can rebalance the things to where they should be. The dysbiotic microbiota is transferable to wild type mice, in which it suppresses inflammasome. So if we can actually suppress activation, so if we can suppress the activation of inflammasome, then we're basically, increase the actual size. So people can read that part. So I'm reading it and they should be able to see it. Do apologize for that. So the dysbiotic microbiota is when they transfer to that, they actually notice that it can actually cause a suppressive effect. It dampens interleukin-18 production, which is a protective effect when it's properly regulated, because it actually pushes up AMPs, which are the things that keep things under control, decreases AMPs production, which is not a good thing, and increases the severity of colitis. Interesting, this phenomenon can be reversed by interleukin-18's injection. So when you inject interleukin-18, as I put up there, you actually reverse this whole problem. And we know interleukin-18 is downstream. It's basically, interleukin-18 is here. So when this increases, AMPs increase and they deal with this problem. But in order for this to increase, inflammasome is required to function. And the thing that actually runs inflammasome is basically our dear taurine over here. That is driving inflammasome. Basically, what we're talking about here, eating a steak a day will make sure that your inflammasome is working properly. your production of interleukin-18 is working properly, that will support AMPs, and those AMPs will keep your gut microbiome in check from going in a deranged situation. That is what a species-appropriate diet does. That is slam dunk. That is basically like fisting a vagoonerized idiot out there that wants to disagree with this mechanistic proof that basically a species-appropriate diet will correct your issues. And we know that because 85% of all vagoons fall out of the diet, and we know why they fall out of the diet is because of major gut issues, because they're not getting enough taurine in their diet. Surprise, surprise. And when basically vagoons start eating some meat, magic happens. All of a sudden, their gut dysbiosis starts going away. They feel better, less bloating, less, because less pathogenic bacteria and all that, causing a lot of bloating and a lot of other discomfort and problems. Things start working better, let alone, I mean, obviously some people, when they may have a long period of inappropriate eating and may have caused other problems like, issues with their pancreas, issues with their basically, liver, kidneys, gallbladder, all these sort of things. And obviously it will take time for some people to sort things out. But if you take a look, and I've talked about it quite a bit, taurine is in every single one of these, fixing up the fibrotic tissue and improving the liver function, fixing up your gallbladder by emulsifying the bile and giving you better quality bile that cleanses the small intestine and reduces and removes the problem of SIBO. Plus, in the stomach, it improves the acidity levels, which means you can digest better by bringing down the pH and increasing the acidity levels, so better absorption and reducing malabsorption issues. And also in the other tissues, they can actually be cleared out of fibrotic tissue and actually work better. Ultimately, as I've said, it is when I say taurine, I mean seafood and meat from ruminant animals. That food, when you put it in your mouth and it goes through your digestive tract, over time, it fixes things. It restores your proper gut functioning. And that's what this mechanistic proof is showing quite clearly. These pathways are affected directly. You take away this sort of stuff, you see this problem happening. You take away the interleukin-18, you reduce it, and downstream AMPs, very small, they drop off quite a bit. By suppressing the inflammasome, you get the inflammasome going inside the epithelial cells, then you basically improve all this other stuff to work. You increase interleukin-18, you increase the AMPs, you get the gut to work again in a proper ancestral way. So from now on, you should be saying, a steak a day keeps the gut doctor away. Anyway, hope you enjoyed that. See yous.